Okay, just to make sure it's understanding B right for 4B. Well, I'll figure it out. I don't think it's too bad. C will be, I probably won't explain very well, but it's fine.
Do your best.
I know, I'm like, I feel like all of this is kind of like, okay,
Well, we're
just do
going.
our best.
It's recording, so.
Okay, so number one for A, we have to see what the banding pattern looks like. And we have to GTP, DD, AD, DD, TTP, ND, CTP. And so we have to draw these out. So I have on one side the DNA sequence that we want to use, which students actually use the same. So for DDGTP, you want to put a band every time there's a C. And then the other one, DDATP, you want to put it wherever there's a T. DDTTP, wherever there's an A. And then DDCTP, wherever there's a G. And then they're each in columns by each DD and TP. For B, suppose I use E. coli DNA polymerase 3, the sequencing reaction to attack polymerase. And fail because the E. coli DNA polymerase is not able to withstand the high heat of PCR like the attack polymerase. And then C, you accidentally add E. coli DNA nuclease reaction in addition to the normal sequencing reaction components. So in the case, we'll split the DNA strand apart, but in PCR, this is already happening because so the reaction will merge, which is an extra helicase. It's not. Might separate it or he separate it too. So it doesn't matter. And then suppose I add DD8 to one of the 4 tubes. However, I forget to add the other DD and ND is just 2 other tubes. Everything else total. So I said the tube with the DDHP will have much shorter strands than the other tube because that is what ends the strand. And the others just have some strands because they're going and not being stopped by those DD entities. Okay, so for number 2.
Okay, so for number 2,
Typical PCR cycle.
during a typical piece, the synthesized DNA
That's at 72 degrees.
while the
Primers and nails complementary
strand
strand
step is done at 55 degrees Celsius. For B, the common selectable marker found on many plasmid vectors is AMP to the R, while the recognizes and cleaves to DNA molecules at palindromic sites. For C, it says during agrogel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive end during electrophoresis, whatever that word is. The DDATP lacks the 2' and the 3' OH group, which makes it terminate, the strand terminate. E, the term used to describe the process where bacteria take up DNA molecules from the environment is transformation. And then F, during blue-white screening, IPTG is commonly used to induce expression of the lac operon, while X-gal is cleaved by B-galactosidase to turn E. coli cells blue.
Okay, so for number 3, A asked to write the sequence of the primers, and you have to do one of the bold ones. So up at the top, the first line, the primer starts on the 3' end, and that goes to 5'. And the first part is going to be reversed because the 3' is over here. And then you just write the complementary ones. So you start at the end here with G, and you do C, A, you see that C, A, G, T, G, C, A, T, T, G, A, G, G, C, and T. And then on the second part, the 3' circuit here, so the primer would start doing the complementary, the strand going in the correct direction, to the forward direction. So you would have start with the C and go G, A, G, C, C, T, T, C, A, C, T, C, A, G, T, C. So that's for A. B is just plugging into the equation. So for the first one, you take, you count the number. So this was from part A. So you count the number of C's and G's. So the first strand, there were 8, and there's a total of 16 in the primer sequence. So you would do 8 divided by 16, which gets you 50% of GC. So you would plug that into the equation where it says percent GC. You got 81.5 plus 0.41 times 50, which is percentage, minus 675 divided by 16, which you get from the total sequence, that's N. And you get 59.8 degrees Celsius. And then you do the same thing for the second one. You just take this strand up here and add up the number of G's and C's, and you get 9 out of 16, which is 56.25%. And then you plug it into the equation, and you get 62.375 degrees Celsius. C asks, let's see. Oh, it says that if DDCTP is accidentally included in the PCR experiment, what happens? So for the first strand, you would start sequencing after the primer. So the 3 A's. So it would be T, 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 A, T, and then it would stop because the next one would be a C, and the DDCTP stops the C's from working. So that's the first one. And then the second one, you come from this direction, and you start at the primer at the C's. So you would have G, 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 A, T, T, A, and then there's D, and then after that it would be a C. So you stop there. So that's right here. So, and then D just asks why you wouldn't include DNA A and DAM in the PCR experiment, and that's because they would both, the protein in them would denature during the heating step.
Four and she number 4 for B and C. But part A supposed to talk about you're supposed to draw the agarose shell from the that was given above. And so we have the first one is BAM HI Eco RI is the second one. The third one is hint 3. The fourth one is Eco RI and BAM HI. The fifth one is hint 3 and BAM HI, and the sixth one is hint 3. So that includes 100 PB, BP, 200 BP, 300 BP, 400 BP, 500 BP, and 1000 BP. But till after I did it, so I have on mine. So I circled the ones that mattered. But this is what we got for cell, and generally what it should look like. You just with the information given to you. So we know that the first reaction has 50, and then 350, and then 500. The second one has 200, and then the third one has 300 and 600. The fourth one has 50, 150, 200, and 500. Fifth one has 50, 100, 250, and has 100, 200, and 600. And so that is what we got. And then going over B, trying to explain it, but the flow that it has 50, 500. So we put those on the line. And then the second, we know it has 200 and 700. And then the third reaction we know has 300 and 600. So then things start getting a little different when you combine them. So we know that we need 50, 200, and 500. We need to figure out where the 150 and 200 go because of the 50 and 500. And the 150 goes to 200. And the fifth reaction is the same thing. 250 ended up going first before the 100 due to placing. And then the sixth one, we ended up with 200, 100, and 600, and the two before the 100. And then for part C, you're using all three of them. And so this was the map we got it based off of what we learned from her video. You got 50 with it's high, and then 100, and then Hind 3, and then that got 1, and then Bam HI, and then we have the 500.
All right. For number 5, you are interested in studying a gene called your favorite gene that you have identified in yeast Saccharomyces cerevisiae. Detail the steps that you would use to generate cDNA from Saccharomyces RNA. Clone a cDNA version of your favorite gene into a plasmid vector and use blue-white screening to identify plasmids that contain your favorite gene cDNA. So this is an outline of our steps. I'll just go through them with you and why we chose them. So the first thing we need to do is we need to generate cDNA from a mature mRNA. And we just use the reading to come up with the steps of those. The first step is 1, purify mature mRNA from the Saccharomyces. And then we mix with poly-D-T primer, which is exactly the mRNA, and reverse transcriptase and DNTPs to synthesize the strand of DNA from that RNA strand with our primers. For 5, we add 1 from E. coli to synthesize the second strand of DNA. So now we have our CA double-stranded cDNA. Next, we want to clone a cDNA version of your favorite gene into a plasmid vector. So the first step in that is we use a restriction endonuclease to cut out your favorite gene from the chromosome it's in. So it cuts it up and then it's digested. And then we add ligase ends on your favorite gene and the plasmid, they'll have some ends that bond and stick together, and the ligase will come in and covalently link those so they're one entire plasmid. So then now we have our vector. So we're going to add IPG and XGAL. What IPG will do is it will start the synthesis of operon. So XGAL will be chopped, or it will act on XGAL. It will lyse it, I think, and that turns the cell blue. So anywhere where the operon will have a blue, and we're thinking that your favorite gene was put in between the lac operon, so it's not synthesized correctly. And so when it's not synthesized, the default color is white. Ones have your favorite gene inserted into the plasmid, and that's how we use blue and white screening for that step.